Hello, everyone, and welcome to ISAAA's outreach program. This is our webinar series on Gene Drive, and this is the third series of Gene Drive that ISAAA has been organizing. I'm the host for today. I'm your moderator, and I'm Dr. Mahalakshmi Arujanan from ISAAA, the Global Coordinator. And I'm also the Executive Director for Malaysian Biotechnology Information Center. You can just call me Maha. So Gene Drive is an emerging technology, as we all know. And because it's an emerging technology, we feel that we need more informed debate, and also debate that revolves around science and not based on emotional or based on misinformation or disinformation. So that is the reason ISAAA has been organizing the webinar series together with Outreach Network for Gene Drive Research. This webinar series. So previously from 2021, we have been organizing on the 101 of Gene Drive. What is the technology? What is the science behind this? And also the applications of gene drives, and what are the potential of gene drives? What are the risks and benefits? Today, this year, what we are going to do is we decided to make it country specific. And we all know Philippines is one of the leaders in Asia when it comes to genetic modification, gene editing, and also they have got very good policies, which is like, I would say it's future-proofed, and also that is science-based. So we are today looking at Philippines. What are the challenges in Philippines that may have potential that can be solved by through this gene drive technology? So with us today, we have got four speakers. Two from the Philippines, and our first speaker is Dr. Nina Gloriani, and she's going to present major health challenges in the Philippines, the infectious disease, especially the insect-borne diseases. And as I said, the gene drive technology is not really here in Asia, but we are looking at the current challenges and how gene drive can play a big role in addressing this challenge. So after Dr. Nina Gloriani, we have Dr. Carmelita Villamo. She's also from the Philippines. She's from Ecosystem Research and Development Bureau as a Chief Science Research Specialist, and she will be talking about invasive alien species in the Philippines. And as we know, gene drive, now for the time being, it has got huge potential as a control mechanism to control invasive alien species that can cause huge damage for biodiversity and even in agriculture, and also for vector-borne diseases. So these two challenges that currently is faced by Philippines, it can really set the tone on how gene drive can be adopted. Then after that, we will take a deep dive into the gene drive technology itself. Our first speaker who will speak on gene drive is Dr. Brian Tarimo from Ifakara Health Institute from Tanzania. He's a research scientist, and he will speak on can gene drive help eliminate vector-borne diseases. Our last speaker, but not least, is a scientist from Adelaide, University of Adelaide, Professor Paul Thomas, the director for the Genome Editing Program, and he will be talking about the diversity aspect, fighting diversity loss with gene drive tools. So this is the introduction to the webinar, and now I'll also tell you the house rules that this webinar, certainly we want to make it as interactive as possible. So what we want from the participants is please share your questions, not in the chat box, but please type in your questions in the question and answer section, which will be easier for ISAAA team to moderate and send it to me. So remember, send it to the question and answer section, not the chat box. And keep your microphone muted, your camera switched on, so that we have a very good bandwidth, so that we can look at the audio and the visual works very well. And also, many of you will be interested to know if you are given a certificate. Yes, all of you will receive a certificate, but when you fill in the evaluation form, you will be given a certificate. So when the link is up, please copy the link and fill in the evaluation form, so that will really help us to improve our webinar series in coming future webinar series, especially in terms of content, because we find this technology really potential, and ISAAA has always been an enabler in adoption of technology. So with that, let us start the webinar, and I would like to invite the first speaker, Dr. Nina Gloriani to speak about the health challenges in the Philippines. Dr. Gloriani.
Yes, good afternoon, good morning, good evening. Thank you very much, Dr. Maha, and thank you to ISAAA for inviting me here. So I will just share. Oh, wait. It should be on the first one. Okay. So I have been tasked to present the major health challenges in the Philippines and maybe focus on later the vector-borne diseases with gene-drive possibilities. So I'm showing here the 2020 statistics that we have for the top 10 leading causes of morbidity here in the Philippines. If you notice, 7 of the 10 causes of morbidity were infectious diseases, upper respiratory tract infection and lower, and we have urinary tract, we have pneumonias, of course, that's lower, we have diarrheas, bronchitis, and TB, all forms. Now, in terms of the top 10 causes of death in the Philippines, this is 2022 data. This time, we only have 3 infectious diseases in the top 10. We have 7 non-communicable diseases that are causing the death here in the Philippines. Now, this is data from the WHO, where it shows of the infectious and parasitic diseases, we have mostly TB in all forms. Then we have diarrheal diseases. Then the green one is tropical cluster diseases. And this red one here is a vector-borne disease, dengue. I'll go back to it later. So the rest are just the HIV, STDs, and so on. So there are 2 vector-borne diseases here in the Philippines that have given us so much problem and challenges, especially for malaria. But this, for many decades, I think, not just years, but decades, malaria posed so much public health problem here in the Philippines. If you note here, this is all red here and way back before 2003, but we joined the malaria elimination program. So from 2003, where you have most of these red provinces here mapped, we now have in 2014 a few provinces in the red. By 2022, that was just last year, we have already 15 provinces with 0 indigenous cases. So these are the green ones. Really malaria-free are the really dark green one. And then 0 indigenous cases, the light green one. And only Palawan remains to have a local transmission of malaria. Now, another vector-borne disease that is now being discussed in many conferences in pediatric, adolescent medicine, and even the adults is dengue. So this is caused by Aedes species. So just to show here over from for so many years, 2010 to 2022, it looks like an endemic thing. And sometimes we do have the big epidemic, that is the outbreak in 2019, and right now this is 2022, but we are seeing a little more cases at the same period this year. So if you see this ASEAN map, now there are many things we'd like to be number 1, no, but we do not want to be number 1 in dengue. So here we are number 1 based on the dengue cases reported from January 2023 to August 2023. Highest number of cases observed here in our country at 80,318, followed by Malaysia and then Thailand. Just to say that among the vector-borne diseases that are transmitted by Aedes, we also have chikungunya, Zika disease here, and Japanese encephalitis. So I decided to include a non-vector-borne problem also. This is actually a global problem, antimicrobial resistance. And if you look here, among the 10 threats to global health in 2019, AMR, antimicrobial resistance, ranks number 2. And in the same year, in 2019, the Philippines had the 128th highest age standardized mortality rate per 100,000 population associated with AMR across 204 countries. I will end with this last slide here, just to connect the AMR with the, at least I found this one publication where a pro-gene, a pro-AG gene drive system for the bacterium E. coli was found to inactivate an antibiotic resistance marker. So we are hoping this could also be used, gene drive could also be used for this AMR problem. Then another one on viruses, we have the virus gene drives. They studied the herpes group of viruses, herpes virus number 5, or this is the cytomegalovirus, where they found the successful transmission by gene drive sequence between distinct strains of this virus, and that this gene drive can efficiently target and replace wild-type populations in cell culture experiments. So at this point, it is in cell culture experiments, but what it shows is a promise that a viral gene drive can be used as a strategy to suppress viral infection. So I'll end my presentation there, and thank you very much.
Thank you, Dr. Nina Gloria. Now, Dr. Nina is a public health expert, and it was so interesting to see that not only vector-borne diseases could be addressed by gene drive, but also viral and bacterial infection as well. So we will discuss that later. Now, before I call upon the next speaker, it's really nice to see the diverse countries that's participating here. Now, I think we have got about 138 participants right now, and we have participants from Tanzania, India, Germany, Philippines, Thailand, Mauritius, Pakistan, Australia, and Switzerland, Kenya, so Ethiopia, Nigeria. I hope I'm just looking at Greece. I'm just looking at the chat box. Now, this really shows that there is so much of interest in gene drive globally, and there's a lot more we could do in terms of engagement so that the emerging technology can take off and really benefit us, especially in addressing global challenges. So with that, I would like to invite the second speaker, Dr. Carlamita, and she will speak now about the invasive species problem, invasive alien species problem in the Philippines. Dr. Carlamita. Dr. Carmelita, you are muted. Can you unmute yourself?
Okay.
Thank you. Yes,
no,
we hear.
Okay. So okay, good afternoon, everyone. So thank you, ISAAA team, for giving this chance to share with you some information about invasive alien species in the Philippines. Next slide, please. Okay, so I was told to give a very simple definition of invasive alien species, and early in this presentation, I would just like to cite the definition of the CBD and the IUCN on invasive alien species. These are species, subspecies, or lower taxon introduced outside its natural past or present distribution, including any part, gametes, seeds, or propagules of such species that might survive and subsequently reproduce. So that's CBD's definition. And on the other hand, the IUCN describes invasive alien species as animals, plants, and other organisms introduced by man into places out of their natural range of distribution, where they become established and dispersed, generating a negative impact on the local ecosystem and species. So if you will look at this very simple presentation, we have an alien species coming to a particular country. When they become established, whether they stay localized or they spread in other areas, but does not generate any impact at all, that is not invasive alien species. But when they cause an impact on local biodiversity, or impact on the soil, or impact on the overall environment, then that is what we call invasive alien species. I would like to emphasize that not all exotic or alien species are invasive. And for an alien species to be invasive, it must successfully outcompete native organisms. They should spread through its new environment and increase population density and harm ecosystem in its new environment. Next slide, please. Okay, so what makes a species invasive? These are some of the characteristics of invasive alien species that we have. They're capable of rapid reproduction and growth. They have high dispersal ability. They have phenotypic plasticity, or the ability to adapt physiologically to new conditions. And they have the ability to survive on various food types and in a wide range of environmental conditions. And why are we so concerned about invasive alien species? When they already cause impact on biodiversity, such as decline or elimination of native species through competition, predation, or transmission of pathogens, and disruption of local ecosystems and ecosystem functions. Next slide, please. Okay, I have just collected some pictures just to give you an idea of the invasive alien species in the Philippines. Maybe it's worth mentioning that the country has already identified 84 species of plants and animals that are considered as invasive. And among the plants that we have, and I'm sure many of you will be very familiar about this, we have the Lantana camara. They were introduced as ornamental plant, but they are now very widespread in our environment, including forests. And then we have a skyflower, or Thunbergia grandiflora, which is very much present here, even in Mount Makiling area. And they were also introduced as ornamental plants. And of course, we would not know water hyacinth, which is very, very invasive at the moment, and the control measures are very difficult to do. We have Hagonoy or Cromolina adorata, and then we have Ipil-Ipil, although we still continue to plant them. They are very invasive and have caused negative impact on our environment. And again, another one is Buyo-Buyo or Piper aduncum, which has been the subject of our research about 5 years ago. And this is very, very prevalent in Mindanao area and even in the Visayas region. And then Mile aminitude or Micania micrantha. And this very beautiful, supposed to be very beautiful tree, African tulip, which has been used in many parks, but they are very, very invasive and has been listed as very invasive even in other countries. Next slide, please. And then these are just some of the animals that we have in the country today, also listed as invasive in the country and elsewhere. Of course, we have the African catfish. Of course, they make good food, but they have the tendency to eliminate even native species. American bullfrog. We have the knife fish, very prevalent in Laguna de Bay and in adjoining Pasig River area. And golden apple snail, which is already a pest in rice cultivation in the country, although they were introduced to serve as food for the farmers and local folks, but they are now finding it difficult to eliminate this in rice areas. And then, of course, we have the janitor fish, although they don't feed on small fishes, but they tend to burrow on river beds, and the problem is destruction of the environment. The Chinese soft-shelled turtle, which is already spreading in many of the freshwater areas, not only in urban areas, but even in rivers outside of Manila. And then the cane toad, which is supposed to be introduced for our sugarcane areas, but are now invasive throughout the country. And gray squirrel, which is found in many urban areas now in Metro Manila. This is a problem. And of course, the mosquito fish, which was initially introduced to control malaria back in the 50s or 1960s, but has proven to be not effective at all because of their limited range of habitat. There's still many more invasive species, and the DNR has a database on this species. So next slide, please. Okay, so the control methods are varied. Actually, for smaller areas where they are present, for instance, in agricultural areas, farmers resort to manual extraction of invasive weeds. And in bigger areas, they do mechanical extraction. Use of chemicals have also been resorted to in some areas, but sometimes it's not good for the trees and for the crops, and even for some animals. And we have biological control initiated in the past, but also which are not effective as well, because some of the biological agents themselves become invasive. And then we have some farmers practicing cultural management practices such as grazing, but this is only good for smaller areas as well. Although the best option could have been prevention of the entry of this invasive species, this has not been done because several entry points, both accidental introduction and purposeful introduction, have not been monitored at all. Maybe next slide, please. Just to okay. But you know, since this is a very big problem in the country, next only to habitat destruction, the DNR has come up with an alien species management strategy, which we call the NISAP. And this strategy actually combines several actions, such as starting with prevention, early detection, eradication, control, and restoration. And we believe at the DNR that this is an effort that should be undertaken with a strong policy and institutional support, with strong leadership and coordination, suitable research and information management, education and public awareness, training and capacity building, and international cooperation. And this is the road to 2026 of our National Invasive Species Strategy and Action Plan. And as party to the CBD, the country upholds its commitment to protect our biodiversity and to address issues pertaining to invasive alien species. And this seminar on gene drive, I look at the gene drive technology as a possible option for the eradication, control, and restoration. Although, as our moderator said, that this is something new for Asia, but maybe this is something to look forward to with proper coordination with the appropriate government agencies and research institution. I think this can be done, and hopefully this can be another control method for invasive alien species in the Philippines in the future. So that's my presentation, and thank you, everyone.
Thank you, Dr. Carmelita. Yes, this is certainly something that Asia should look into. The only problem is all your invasive species looks very pretty. And I think that is why people started putting it in the ship and bringing it to other places.
Yes.
Many countries is facing a huge problem here. And now I would like to also bring to attention that we have 165 participants online. And this again shows the need for information, science-based information to be disseminated. And I would like to welcome my colleagues from Malaysia, and we have from other countries as well now. So welcome everyone again. So our next presentation, we'll go into the real actual topic, and this will be the first speaker for the gene drive itself is Dr. Brian Tarimov from ISAAA Health Institute, and he will speak about vector-borne disease. How can we eliminate vector-borne diseases using gene drive? Dr. Tarimov, the screen is yours.
Thank you, Maha. Good morning, good afternoon, good evening, everyone, depending on where you are in the world. Let me share my presentation, and I'll give a short but succinct presentation about what gene drives are and how can they be used for control of vector-borne diseases. So what is a gene drive? In normal Mendelian inheritance, any offspring has a 50-50 chance of inheriting a genetic trait from either of its parents, either from the mother or from the father. However, what gene drive is where this genetic trait or the genetic element is able to bias in inheritance. And therefore, instead of having a 50-50 chance of inheritance of this gene, there's a near 100% inheritance of the trait. So you can see on the left, the normal inheritance, there's a 50-50 chance of having inheriting a particular gene from either of the parent. But on the right, where there's a gene drive, the genetic element in the yellow, it's been able to bias itself, and therefore in the offspring, it's prevalent in all of the offsprings. So how does this appear in terms of our population? So on the left, we have Mendelian inheritance. The circles in yellow or in orange, you want to pass a specific genetic trait across your population. And you can see from one generation to the other, from first generation, second generation, up to the nth generation, there's a 50-50 chance of inheritance of this genetic trait. And therefore, your genetic trait does not, its frequency in the population does not rise, and it remains more or less the same. Now with gene drive on the right, you have because this genetic trait is able to bias its inheritance, and therefore you have its frequency increasing from one generation to another. And therefore, as generations go on, the frequency of this genetic trait is able to increase into the population until a level after several generations where it reaches fixation, and therefore you've been able to pass this genetic trait across your population. Now, how does this work? Sorry, I'm trying to go to the next slide. So how does this work? The way this works is that if you can, there are specifically types of genes in our genome calling homing endonuclease genes. And when these genes are expressed, they produce this RNA, which it's able to recognize the homologous part of the other of its homologous chromosome, and it's able to digest it. Now, the cell has an inherent repair mechanism whereby this cut chromosome has to be fixed. One way of fixing it is it could join the two ends together, and then, or it could use the template DNA, the template chromosome, and use that to fix, to repair the other chromosome, and therefore in that way you have the gene of interest being copied into the other chromosome. And if you restrict this type of repair mechanism or this type of work to happen in the reproductive organs, either in the egg or the sperm, you are ensuring that your gametes will have a copy of each of the gene that you want to, or genetic trait that you want to pass to the offspring. Now, gene drive is a natural occurring system. They do occur naturally in nature. And what science has done is to be able to adopt some of these, what's happening in nature, and into synthetic gene drives for to use for the control of vector-borne diseases. So some of these naturally occurring gene drive system include sex chromosome drives, transposable elements, homing-based gene drives elements, which I just explained. There's the media and the dominance, and also cytoplasmic incompatibility, what we see in the Aedes mosquito and Wolbachia for the control of dengue. It's some form of a gene drive system. Now, what are the current applications of gene drive? There's quite a few applications of gene drive. There's application of gene drives in human health, environment, agriculture. But what has been mostly extensively researched up to now is the control of invasive alien species, and also for the control of vector-borne diseases. And I will briefly talk about gene drives for the control of vector-borne diseases. And my colleague Paul, who will come after my presentation, I think he'll talk about a little bit more about invasive alien species. Now, why vector-borne diseases? Diseases such as leishmania, malaria, dengue, chikungunya are all transmitted by mosquito vectors. 80% of the world is at risk of vector-borne diseases. The burden, almost 70% of the global burden of communicable diseases is due to vector-borne diseases. And there's over 700,000 deaths annually because of vector-borne diseases. So you can see it's a massive burden on the globe, and something needs to be done about it. This slide here shows a number of indigenous cases, imported cases, and outbreaks that happened between 2017 to 2019 across the globe. And just looking at these 5 vector-borne diseases: malaria, chikungunya, dengue, yellow fever, and Zika. And you can see all across the globe, especially in the temperate, in the subtropical and tropical countries, the burden of vector-borne diseases is quite extensive, and something needs to be done about it. Now, what has been shown that if you can control these vectors, the mosquitoes, the fleas, the ticks that transmit these diseases, you more or less have been able to control the transmission of the disease they transmit. So on the left here, I have a brief history of what vector-borne diseases looked like. And before World War II, extensive work was done on environmental management in order to control mosquito vectors. However, after World War II and the discovery of DDT, this is what led to a lot of spraying and led to eradication of malaria in the US and some of the countries in Europe. And then in the 60s came the Global Malaria Elimination Program, which also extensively used DDT, but then failed in the late 60s. And then ITNs were introduced in the 80s. And then from the early noughties, the early 2000s, there's been a high extensive use of long-lasting insecticide-treated bed nets, LLINs, and indoor residual spraying and other tech, and also use of drugs and effective diagnostic tests in order to control these diseases. In my next slide, I'll just briefly use malaria as an example. If you look from the year 2000 to the year 2015, and the black line on the top shows that if there were no any intervention being used to control malaria, and then the red line is what actually happened, the actual reduction in malaria that happened. And you can see the green part, which is attributed to ITNs, to the bed nets, contributed significantly more compared to other interventions against malaria. And this shows that if you can effectively control the vector, you pretty much can effectively control the transmission of this disease and also perhaps lead to elimination. However, there's a caveat here in the sense that some of these control tools or control interventions are not successful. We have lack of specificity for ecosystem damage because the insecticides are not they killed indiscriminately. They just don't kill the vectors. They can kill also other useful vectors, the butterflies, the bees, and which are useful to agriculture and other forms of economies. What do we do about those mosquitoes that they do not bite indoors because you sleep under bed net inside your house? What happens when you're outside the house? And there's some habitats that are difficult to reach. So in order to do environmental management, it becomes a bit tricky. And fogging and fumigation that has been used to control some of the Aedes mosquitoes and other mosquitoes, there's toxicity, and also there's some quite of nuisance with them. So this, with all of these, challenges with regards to vector control, it has led to scientists and other researchers in the vector-borne diseases sphere to think beyond what can be done besides some of these traditional vector control tools that have been done. And in doing so, scientists have been able to look at what makes a mosquito such good vectors. There are certain factors that if you look into the mosquito, they enhance their vectorial competence, their vectorial capacity. The host feeding behavior, why they prefer, there's the human biting preference, the vector susceptibility. That means the parasite, the pathogens that they carry have to survive so that the mosquito is able to transmit from one individual to another. the longevity of the vector. We know the mosquitoes have to take at least 2 blood meal in order to transmit the pathogens they transmit. They have very high reproductive rate. One mosquito could lay over 100 eggs in one go. And then the habitat preference and so forth. So if we look at all of these factors that make mosquitoes such good vectors, we can pretty much say confidently that they have a genetic component. And if they have a genetic component, it means therefore we can identify the gene, we can modify this gene in order to prevent these mosquitoes or to be able to be good vectors for transmitting the diseases. So in the mosquito genome, the genomes for Anopheles, Aedes, Culex, and other vectors that transmit diseases are out there. They're in the public domain. You can identify genes that are responsible for longevity. You can identify genes that are responsible for its immunity. So therefore, you can be able to block development of parasites, reproduction, and feeding behavior, and so forth. Therefore, how is this done? Therefore, once you have identified these genetic characteristics, you modify your mosquitoes, and therefore you release your mosquitoes. And on the left here, the first approach is to do population suppression. Therefore, for example, you have genetically modified mosquitoes' reproductive ability. So therefore, once you release your mosquitoes, which are the ones in gold here or yellow, over time they'll be able to over-dominate the wild type, which is the one in black. And after some time, you have that population will not crash, but it will go down to such a level that transmission of your vector-borne disease from one individual to another will be extremely unlikely. And another option on the right here is population replacement or modification, where, for example, you've been able to improve these mosquitoes' immunity, and therefore, when they carry a pathogen, the pathogen is not able to fully develop in the mosquito, and therefore, when this mosquito go and bite another individual, they're unable to transmit this pathogen. And therefore, so if you can see on the right, after some time, your wild type population will reach to a level where they will be over-dominated by your transgenic mosquitoes. Now, before I go to this slide, there's one thing I forgot to add here, but it's important worth mentioning, is that what is so good about this genetic modification mosquitoes as a vector control tool compared to what is currently being used here. And I'll just stick to 3 points here. The first thing is the transgenic mosquitoes will be efficient. Why they'll be efficient? It's because your genetic trait will be transmitted from one individual from parents to offspring through sexual reproduction. And therefore, you do not need any human component to ensure that your intervention is effective. What do I mean by this? For example, with the bed nets, for example, you give it to individuals, you have to ensure they sleep under bed net. You cannot go into their houses and make sure that they sleep under bed net. So you are trusting them that you're giving them the bed nets, they will sleep under it. But with transgenic mosquitoes, you just release your mosquitoes, your genetic trait will be passed on to the population, to the next generation through sexual reproduction. You're sure that these mosquitoes will be able to mate, and therefore the efficiency of your intervention is nearly 100% compared to other current interventions that require the anthropogenic human intervention in it. Second point I'll talk about is long lasting. Long lasting is your trait is going to be, with some of the interventions that we use right now, after some time they need to be replaced because, for example, bed nets, they get worn out, they get torn, the insecticide in them gets depleted, and then you have to do another massive campaign to give these bed nets. But with your mosquitoes, once you release, and that's it, because of the gene drive, you're ensuring that each population, each generation will get the genetic trait that you want. And also they're easily accessible. a lot of the interventions that we have right now, not only for vector, even if you look at the human, to control the parasite in humans, the drugs, the diagnostic kits, and so forth, they require a lot of logistics. You need to transport them to an area, you require a warehouse for storing them, you require vehicles to transport them from one area to another. So there's a lot of logistical challenge which come with a lot of financial burden. With your transgenic mosquitoes, what you do, you just release them and they will do the work for you. Now, I'm not going to say here that the gene drive mosquitoes for vector control is the silver bullet, is the panacea. What we're trying to say here is that this technology, in complement with what's being used now, can really, really bring significant impact in the control of vector-borne diseases. However, there are certain issues that arise with gene drives. There's resistance. Like any other intervention, we've seen resistance in insecticides, we've seen resistance in drugs, we've seen resistance in antibiotics, vaccine, and there's also a good possibility for gene drives to develop the resistance. Therefore, more research is required to ensure that the less likelihood of building resistance before this technology is released. There's ecological and biological diversity effects. So a lot of groups right now working in gene drives or transgenic mosquitoes with gene drives are looking into, are mosquitoes a pioneer species in the ecosystem, in the ecological niche? What happens if it's potentially these mosquitoes or are removed from the ecosystem or from what happens to other organisms? Also, the containment. How do you ensure containment, especially during the research stages, so that that these mosquitoes do not escape and bring any harm to the surrounding areas? And also, there needs to be community acceptance and also regulatory acceptance that's international and able to effectively regulate the technology. Now, all this being said is that there is phased, the technology there isn't any gene drive mosquitoes released right now anywhere in the world for vector control. What's happening right now, it's all this technology is still in the laboratory stages or in small cages. So what happens is,
do you have is this your last slide?
Yes, yes, I'm done. I'm almost done.
Okay.
So this is the stages. We go from the laboratory, then the small cages in contained spaces. Then you move to large stage cases, and then you do the release. And each stage you gather results, you gather information that will be able to tell you should you go to the next stage or not. So it's not like you just go from one stage to another. Each stage has like a gate where they like, should you proceed or not proceed? There are certain groups in Africa working on gene drives. I've just shown a few here. There's a group in Burkina Faso, Mali, Tanzania, Kenya, Uganda, Sao Tome and Principe, Comoros, and so forth. And this is my last slide, that these gene drives have great potential for the control of vector-borne diseases. And thank you very much. I'll stop here.
Thank you, Dr. Tarimo. I think you have nicely given a very comprehensive overall picture, and also you even gave us the key takeaway messages. Now, because of time, I'm going to go straight to Professor Paul Thomas, who's going to talk about fighting biodiversity loss using gene drive tools. Prof. Thomas.
Thank you, Maha. Hello, everyone. Thanks for joining the session today. I'll just share my screen and we'll get started. Okay, right. Yeah, so as Brian has nicely introduced already, gene drive technology is actually quite well advanced in insects with the purpose of controlling vector-borne diseases. I'm going to take a different tack and tell you a little bit about the potential of this genetic biocontrol approach to fight biodiversity loss, in other words, invasive species, and the progress that's been made in that area over the last few years. Okay, there we go. So in terms of the global impact of invasive species, we actually have a very good recent estimate of the costs associated with invasive species across the world from a report that's shown here on the bottom. And essentially the bottom line, or the figure that's attributed to the cost of invasive species, is around $423 billion every year globally. So it's clearly a huge problem. And what's more, this cost has quadrupled every decade since 1970. So it's a problem that's not only huge, but it's also increasing. There are many types of invasive species. We've heard about some of them already. In the Philippines, I noted cane toads are also a problem in the Philippines. They are in Australia, a considerable problem, where they were also introduced for cane beetle control. And grey squirrels, I also noted, were mentioned earlier, and they are a big problem in the UK, I know. But in terms of invasive mammals, that's really got to be where the focus of today's talk is, partly because of where I work, but also because of the huge costs associated with this particular group of species. In Australia, they cost around 20 billion, or they have cost around 20 billion to the economy over the last 6 decades, and they're a major driver of extinctions across Australia. So we need some technology, some control measures that really address these huge issues that we're facing for the environment. Some examples that really, I think, bring home how quickly invasive species can expand their populations under appropriate conditions. One example of that is the mouse plagues that we have in Australia every 4 years or so. And these are principally in grain-growing regions. And an example of that is shown here. So, I hope this doesn't give you nightmares, but I really just think this kind of footage brings home just how, in what huge numbers mice can breed to really very quickly when the conditions are right. So there's lots of food for these mice in these plagues because of the grain-growing regions, and the environmental conditions allow them to expand in numbers quite quickly. The other aspect of particularly rodents in terms of their invasive and destructive capacity is on islands. Rodents frequently get to islands because of basically they are stowaways on seagoing vessels. They invade islands, and when birds particularly are present on those islands, they cause great loss of biodiversity by eating the chicks, eating the eggs, devastating bird nesting sites, etc. And although it is possible to eradicate rodents on islands if they're relatively small, using traditional sort of baiting technology, this has several disadvantages, and really it's not a landscape scale solution for these really invasive pests. So we've heard from Brian already about how gene drives work, and I just want to sort of recap that concept now and tell you a little bit more about how it's been applied in particularly mammalian invasive species. So we've heard already that essentially genes come in pairs. So the basic law of genetics is that we get one copy of a gene from each parent, and the chances of us transmitting that gene to the next generation is essentially 50-50. It's like having a toss of a coin. It's a head or a tail, and that determines whether either version of the gene is passed on. Well, the gene drives break those rules, or they don't obey those rules, and instead they have an enhanced chance of inheritance. So it's a bit like having a coin with two heads, and that means that the gene drive, the genetic modification, or the particular version of the gene that's been synthetically engineered in a laboratory or is naturally occurring, is transmitted to the next generation with a much higher frequency than it normally would. And what this means, as Brian showed us already, is that a genetic modification or a gene drive can spread through a population actually quite quickly. And this is essentially one way that you can modify a population, which is great, but it's probably not the ideal strategy for invasive species, where really the objective is to eradicate that invasive species from the environment. So instead, you can engineer a gene drive in the laboratory to do an extra trick, and that is to suppress the population to create infertile females once it's reached saturation. So in other words, once gene drive bearing organisms start to mate with each other, they can generate infertile females, and that can cause a population crash. And this kind of strategy has been applied in insects, and I'll show you can also be applied in mammals as well. So the reason there's so much discussion and interest in this area is of three basic features of gene drives. First of all, it's a non-lethal form of population suppression, unlike the baiting regimes that are present at the moment. It's landscape scale, so with just a few organisms, you can cause a population to crash. And thirdly, it's species specific because mice only breed with other mice, etc. So the reason we start with mice as the first port of call for trying to develop gene drives in invertebrates are that we already know how to engineer the mouse genome quite well. So we have the technology to create the gene drives in a laboratory setting. And also mice, as you've seen, are quite serious invasive pests. So it's a good place to start, but we see that there's potential for this technology across the invasive species spectrum. Now, in terms of what's been done over the last few years, you can capture that in this slide. And all I really wanted to say is that, as Brian nicely explained, gene drives come in different flavors. There are different types of gene drives. The one that he described in the most detail is this so-called homing drive, where the gene drive element replicates in the germline by inserting an extra copy into the chromosome. This has been attempted in mice by us and other groups, and essentially it's not working efficiently enough for us to really consider this as a deployable strategy and taking it forward. So we've kind of shelved this one for the moment, but noting that it's working really very efficiently in insects where it shows a lot of potential. There are other approaches where you can look to bias the population towards, for example, a completely male population to cause a crash through lack of females. They're called X-shredders or driving Ys. Again, that doesn't really seem to be working particularly well in mammals as far as we can develop so far, but it's early days. The one I'm going to tell you about, though, is a strategy that we've recently developed in mice, so you'll be pretty up to date by the time you've heard the next few slides. And basically, it's the only version of a vertebrate gene drive strategy or tool that's been proven so far for proof of concept at the laboratory stage. So this is still very early stage development. And essentially what we've done here is to leverage the fact that we can use a gene drive element that's a naturally occurring element that's found in mice in the wild. And that's called the T haplotype. I don't want to, get lost in the weeds too much about the way this system works because it's fairly complicated. But suffice it to say that males that carry this so-called T haplotype will pass it on to females up to 95% of the time. So it's already a strong driving element that we can use to potentially modify it in such a way that we can use it to suppress a population. So that's basically the idea behind this work. And the way that it works is so-called, it's part of a toxin antidote system. And again, we don't need to get lost in the details of this, but I just want to give you a little bit of details because it is quite a neat system. And the way that it works is not dissimilar to some of the other toxin antidote systems that are naturally occurring drives that Brian alluded to in his presentation. And these are found in species all the way through to insects. So basically what happens during sperm development is there's those sperm that contain the T haplotype that's shown here in red are linked to sperm that don't contain the T haplotype. And the T haplotype releases a toxin that gets transported across into the sperm that don't carry the T. And this effectively makes them very poor swimmers. So they can't really, they lose the race when it comes to fertilizing the ova after mating. But the trick that gives the T haplotype its edge is that it also contains an antidote to this toxin that's not shared. So in other words, what happens during mating is that you've got competition between the blue sperm and the red sperm pretty much always win that race because they have the antidote that protects them from that T haplotype toxin. So we looked at this system and we thought, well, maybe we can modify it in such a way that we can turn it into a suppression drive using CRISPR genome editing technology. And the strategy that we devised is shown on this slide. We call it T-CRISPR. Essentially, there are two things that happen, and one of them is already happening by virtue of the fact that we have this naturally occurring drive. So this T haplotype shown here in red. And we know that the T haplotype is transmitted to 95% of offspring from males. And we engineered it in such a way that it would also create mutations in a gene that's essential for female fertility during the germline development of males. So it didn't affect the male germline. They were completely fertile, but we just wanted to create mutations in genes that would ultimately cause female infertility. And I'll show you how that works in a moment. So this idea was kind of a new idea, but we couldn't really get a good handle on how well it would work without doing some pretty sophisticated computer modeling. So I wanted to show you that now because I think this is an important part of gene drive development, and all groups that are developing gene drives have a version of this kind of approach. And essentially what you want to do is to take the best field measures, the best data from the environment, work out a way of incorporating that into a model on a computer, and then ask the question, how would your gene drive strategy work if you deployed it within that type of environment? So that's what we've done here. Essentially, you're looking at 200,000 mice that are included in this model, and they're incorporated into this virtual island that you can see here. The mice that are in black are the wild type mice, and you can see I've just seeded in a few red gene drive mice, just 256 into 200,000. So that's 0.1%. And you can see over a period of time, we get spread of those gene drive mice throughout the population. And then as those gene drive mice start to mate with each other, that infertility starts to kick in, and that we get a decline in the population, a steady decline in the population over time. And by using this kind of computer modeling, we get some confidence that the strategy could work potentially as a suppression drive in mice. So to test that a little further, we actually engineered this concept in laboratory mice. So this is the very beginning stage of that pipeline that Brian showed. And I'm not going to show you the data, but just I will show you here the publication that we put out last year. And if you want to look it up, by all means do. And I'm happy to take any questions afterwards. But what we were able to show is that the concept that we developed on the whiteboard and that we modeled, that I've just shown you the modeling for, we could create that in a laboratory setting in laboratory mice. So we have proof of concept for this T-CRISPR strategy, which is, as I said, the first time we have something that we could point to say, this is actually something that could be developed further, at least for mouse genetic biocontrol. So in the last couple of slides, I just wanted to tell you what can we do beyond mice. So they're not the only invasive species, as we well know. We're very interested in thinking about strategies that could be transferred across different mammals and potentially even different vertebrates. And we wanted to sort of think about ways, or sort of what is the potential of those strategies if we plug them into the model in the way that I've shown you already. So this is an example of the mouse modeling. If we use the same kind of approach to model rat, so some of the designs that we've been thinking about, if we use those designs, but with rat parameters in the model, we see that there's lots of potential for eradication also. And even rabbits that have a longer generation time and fewer offspring are still a pretty reasonable species to think about gene drive applications for. What I would like to point out is that serious invasive species like foxes and cats, which are a massive problem, particularly cats in Australia, they're probably less amenable to this type of technology because their generation time is longer and they have fewer offspring than mice or rats. So again, coming back to Brian's point, we don't necessarily see this as the silver bullet for all invasive species. I think it's going to be an element of some of the biocontrol measures that we can take for at least some species. And those species are probably the ones that have the largest number of offspring and the shortest generation times. They're going to be the most amenable to this kind of approach. So to conclude, our group and other groups have looked at a number of different genetic strategies for rodent suppression. Some of those have been borrowed from insects and they're working quite well in insects, as Brian described. But it's really been a mouse-specific one called T-CRISPR that is the first validated genetic biocontrol strategy for mouse population suppression. And this has been validated only at a laboratory stage at the moment. And whilst this is, you know, a significant start to the journey towards using these genetic biocontrol, you know, tools down the track with the appropriate controls, which I'll come to in a minute, it's really important that we start thinking about how we can use these in a safe way. And what I mean by that is to target their actions so that they're not going to affect the native range of the species. So, for example, coming back to the cane toad issue again, in South America is where cane toads come from. We don't want a gene drive organism to get back into the native range, in that case, South America, to suppress the population where they're part of the normal environment. So we're thinking also about ways to limit the activity of the gene drive so that they're specific for the target pest population that we're interested in. Gene drives have a lot of potential beyond mice. I just showed you a mouse example because that's really where the field at is in the moment, but we're very interested, as I said, in developing transferable strategies to other pest mammals. Probably rats would be the next species to think about there. And lastly, we've had this point already, but I just wanted to underline that really this is very much early stage development at a laboratory level. We are already working with social scientists to do stakeholder engagement. And also the regulators in Australia are very interested in this area, and we're constantly working with them to not only license the laboratory experiments, but to think about how it would, how regulation could proceed in the future if we have successful laboratory trials. So with that, I'll finish up, and I just did want to thank those in my group who've been involved in the study, and they're shown up here, and particularly Luke Giras, who's a PhD student who did most of the T-CRISPR work with Ashigul, doing the modelling Ashigul-Barrand. So with that, I'll finish up and hand you back to you, Maya.
Thank you, Paul. I see that you have got eggs in 3 different baskets, and I see that as we need all tools to address the global challenges, especially when it comes to biodiversity and also diseases, emerging diseases, and the diseases that has been there for a while. Now, we have very good interaction now with the audience. I see a number of questions. Now, since gene drive is still an emerging technology, I would like to go first with the basic question on the concept of gene drive itself. So the question here is how gene drive is biased in terms of its inheritance. Either Paul or Brian, can one of you take this question? Brian? Yeah.
Let me give a crack at it, and then if I miss anything, Paul can add on. And to answer this effectively, let me share something really quick. And maybe this did not come across clearly. Can everyone see my screen now?
Yes.
Now, so we have 2 pairs of chromosomes, or 2 pairs. So we have so if you can look at this genomic DNA here, the red part is going to be nicked off, so it's going to be a double-stranded break, and then you have the gap, and with the gap here, it needs to be repaired. And one way of being repaired, what I mentioned is called homology-directed repair. That means it uses the complement chromosome and then to copy itself. And then what you have is you have the repair being made. And the other option is to use non-homologous end joining. That means it just joins this end, and this is where you have the insertion or deletions. Now, with how does gene drive being bias inheritance is if this template, which you are used to repair the broken chromosome, has your genetic trait you want to spread, that means you're going to have your genetic trait being copied to the other part of the chromosome, and therefore your 2 chromosomes are going to have the genetic trait that you want to propagate. And if you restrict this process to happen in the germline when the formation of gametes, that means you're ensuring that your gametes are going to have your genetic trait that you want to pass on from parents to offspring. So another good analogy, for example, this is a very good analogy that my friend here, Tony, uses a lot. So for example, let's think about you have a very nice carpet. And then with your carpet, you have this part being cut off. Now you can actually sew this part. You can just the end here and the end here, and you have something like this, which is a ruined carpet, or you can use this template here to make this part, and then you have your perfectly nice carpet again. So I don't know if I've been able to answer that question, but the way that the gene drive is biased is that it's restricted to the germline, and therefore one of the template of the chromosome is used as a template to repair the nicked chromosome, and therefore you have a copying of your genetic trait into both of the chromosomes, and therefore all your gametes or all your eggs will have the genetic trait that you want to spread. If I've missed anything, Paul, please add.
Paul, any other analogies?
No, that was great, Brian. I don't have anything to add except that you've described nicely the main mechanism that people think of when they hear gene drive, and that is that homing drive that you put out. There are other versions of gene drives as well, so it's not the only way it can happen, but it's certainly probably the most significant way it can happen, especially in insects.
Paul, the second question is to you. What desirable features of invasive species that makes it, that we can, is a potential candidate for gene drive applications?
Yep. I guess we think about lots of factors when we think about targeting a species, and that's where the modeling comes in. It's very useful because we can actually look at the time it takes for eradication by putting in the best sort of field-based knowledge that we can into those models. So it's cost, I would say cost and time to eradication, and how good the current tools are as well. So it's really a combination of all those things, and also knowledge of the genetics and genome structure of the organism itself. So mice really tick all of those boxes because we know a lot about the mouse genome, we know a lot about the genes, it's an invasive pest, and it reproduces quickly. And it's expensive in terms of cost to the environment and to the agricultural sector. So it's really putting all those things together. If we had another species that was really doing a lot of damage to the environment, but we didn't really have a clue about its genome or what the genes did, that would be a much more difficult exercise to try and build a gene drive to target that organism. Oh, and one more thing, sorry. It also helps if you can propagate the species in a laboratory setting, because it's extremely difficult to develop a gene drive unless you can have access to the species and essentially manipulate its genome and its breeding.
Okay, now that we spoke about the basics, a bit of the basics of gene drive, to the Filipinos experts here, either one of you, Nina or Carmelita, what is the Philippines stand on adopting gene drive technology, and which department will be responsible for the regulations of the environmental release or field trial of transgenic animals that's developed through gene drive? Anything in place already, or is it under discussion? Carmelita, you want to?
Okay.
Yeah.
Yeah. I thought we will not discuss this yet, no, because this is a very even Dr. Paul said that one gray area could be the area of regulation. Now in the Philippines, we have a biosafety committee that will handle this, and this is a group of experts coming from the different agencies. We have the DNR, the DA, and all other agencies involved in genetic modification. So I think it will have to pass through this body before it gets into place, even from the very start of the experiment up to the time it is allowed for use. So it's a very long process, just like what the GMOs had undergone in the past. So I think that's how it should be, and that is how it should be, because we just cannot move on doing this with appropriate research agencies, but it has to go through the process of being done under containment, and then being released to the environment before it becomes really useful in the future. So as I've said, it's a long process, and I think something that has to be discussed very thoroughly before we even think of adopting the gene drive. Although I said that it has quite a lot of potential for controlling invasive species. Not all of them, but many of them that can be subjected to gene drive technology.
Nina, is there anything you want to add on the Filipino situation?
Well, not exactly on gene drive, but there was a time we were trying to use the technology, not really use, but to try the GM mosquito technology. And it was very difficult to get people to study it, especially from the medical and the public health sector. So I don't know about gene drive also tinkering with this, but of course it has its potential.
Yep.
It has to be really studied very well, discussed, consulted through a lot of consultations.
Yes. Talking about it has to be really studied well, and the risk assessment, risk management has to be in place. I'm going to combine three questions together. Maybe Paul and Brian can attempt this. If we eradicate or suppress the population of certain species, what is the effect to the ecology? And also, what are the other the other effects to biodiversity through this biased inheritance? Paul?
Yes, I get this question occasionally. It's a good question. I think it's an easier one to answer for invasive species because those species are not native to the environment. So you're looking to remove something that shouldn't be there in the first place. We know quite well what the effect of removing rodents on islands is through baiting programs that have taken place. There's been hundreds of successful eradications on islands using traditional bait technology, and the results are quite stunning in terms of restoration of the ecology, the bird populations, and even the reefs around the islands benefit because when the birds come back, all of the phosphates that's associated with the guano add nourishment for the reef systems that are around the island. So the reefs flourish, the fish flourish. So at least in that context, we know the benefits are considerable. I'm going to let Brian answer maybe from a mosquito perspective, because I think that's a really interesting question as well.
Thank you, Paul. Yeah, we do. I also do get this question quite a lot. And with regards to mosquitoes, and if you look, for example, Anopheles mosquitoes that transmit malaria, if you look at the whole genus, there are over 3,500 mosquitoes that are in Diptera, in the class Diptera, and then about 400 that are in the genus Anopheles, and then about 70 that have been confirmed through research to be able to transmit malaria. But what I've been describing and what a lot of groups globally working in gene drives for malaria control, they're talking about one or two of these mosquitoes, and it's predominantly Anopheles gambis sensostrictus, which is perhaps the most efficient malaria vector globally. So if you eliminate one, or if you suppress one out of 3,500 mosquitoes, statistically, you can say it's negligible, but still, we should not be playing God here and just trying to eliminate a species. So what these groups are also doing is they are looking at what is the importance of Anopheles in the ecosystem. Is it a pioneer species? Is it a predator, or is it a prey of another species, and so forth? Little evidence has been found with regards to the contribution of this species of these mosquitoes in the ecosystem. What is currently known is the larvae. It's perhaps a type of food to certain fish, but these fish have also been shown to feed on other type of mosquito, not just Anopheles, but Culex and Aedes and so forth. So briefly, and we're not talking about eliminating a species. We're talking about suppressing a species. We're talking about reducing its numbers to such a low point whereby it's unable to transmit the disease. Thank you.
Yeah, so looks like, studies are well underway to look at what is the effect. And also the main misconception is that people think that the completely is going to be extinct. No, we can't do that. It's not eliminating a species. And Paul, what you said about removing what was already not there and getting back to the original ecosystem. Now, what is the problem with that, right? Because we never had that alien invasive species in the first place. So I think this is what needs to be communicated to the stakeholders. Now, moving on from this sort of risk assessment, risk management, the next question is, how do we move forward then? What would be the enabling policies that can fast track the research and future applications of gene drives across sectors? Today, we're talking about health and biodiversity, but also in agriculture. Brian, Paul, since you have experience in this, what are your thoughts on this? Paul, you want to go first?
Well, yeah, it's a big question. I think there needs to be a recognition at governmental level of first of all, that this kind of technology is a little closer than perhaps they realize, in terms of it's something that it's no longer, on the far distant horizon, but it's on the closer horizon. And so I think the first thing is that we need to get better at communicating to policymakers and decision makers about the potential of the technology and opening those channels of communication. I hope that once that process is underway, then we can get more traction in terms of promoting policies and even funding avenues to take it to the next level. Because at the moment, I'm sure Brian could relate to this. We have the situation where we've got a technology that we want to move essentially from the laboratory proof of concept to an enclosure or cage trial that's larger and larger. And to do that, it becomes quite expensive. So at the end of the day, this technology is not going to get towards deployment without having those trials in place. Funding mechanisms have to be there to enable us to actually, make that happen. But of course, all of this has to be done in consultation with the community in the broadest sense, because the last thing we want to do is to get to, the end of it and say, look, we've got this technology, but we haven't consulted with the relevant community. So we're very conscious of that. So risk assessment, stakeholder engagement and regulator engagement are part and parcel of that path forward.
Yeah, I always believe when we build the political will, whether through social licensing and by empowering the people, where public participation is empowered and people make well-informed decisions, or request to the government to move certain technology, I think then the political will is built, and that actually is an enabler for funding and also for policies, policy development and policy implementation. Brian, what do you want to say about moving forward?
Yeah, I pretty much agree with what Paul has mentioned, that there needs to be political will, effective engagement with communities, and also regulation has to be in place. Surprisingly, very surprisingly, because if you look in the whole spectra of genetic modification, the agriculture side has always been the forefront compared to us in the health. We even in terms of regulation, a lot of the things we use, we've adopted them from agriculture, biotech. So, but in terms of gene drives, they have been slower, and I don't know. Perhaps the few people that I talk to, when food is a very contentious issue. People are like, oh yes, we want diseases to be cured, but don't touch our food and stuff like that. So there needs to be better communication from us scientists to the policymakers, the regulators, and even the communities that this technology could actually improve some of the food production in agriculture. Thank you.
Actually, medical biotechnology or vaccine production, drugs have really benefited from genetic modification and also gene editing. But the thing is, the public do not know that these tools are really used for their drugs and for their vaccines. And even when they know, like for example, insulin in 1980s, even when they know, it is just it's more easily accepted than what you said, Brian, the food, because food is like, it's my food, I'm going to eat it. But they do not understand that the drugs and the vaccines and whatever injectable, it also goes into our system. So that definitely I agree with both of you that communication is really important, and that's where ISAAA plays a big role. Now, someone put a very interesting comment here, anopheles, it actually means useless. But then, of course, we still need some risk assessment and risk management and look at what's the effect. Now, the other question here, which I don't think you have to answer, is that what happens if we eradicate population or species? Now, as you said, we are not eradicating a species. It is just, just a small portion of the population, and it's also country specific. But I've got another question here. I think it's an interesting question because today we're talking about alien invasive species, but and also public health. But what about the application of gene drives in microbiological setting? Nina, you spoke about microbiological setting, but then it was still related to health. But are there any other things beyond health for industrial purposes? For environmental purposes, cleaning oil spill, for example, or bioplastics, anything like that?
I think that's possible. I mean, it's less contentious if you're talking about cleaning the environment, bioremediation or something. But for now, for health, I was just looking at, there's a question here, can gene drive lead to antimicrobial resistance? I don't know who could answer that. The question is, I was actually trying to say that it can fix or be an intervention for antimicrobial resistance, no? But the question is the other way. Can gene drive lead to it? I really don't know. Maybe Dr. Brian or Dr. Paul can answer that.
Okay, so we are coming to the end of the session. Now, the last question I have here is based on available evidence for the lab and modeling with regard to vector control gene drive, how long will it take to implement in the field? I think maybe we can go with this question because we know it takes about 15 years. Those days for genetic modification, it is taking a little lesser for gene editing. How about gene drive, Paul and Brian?
Yeah, it's a good question. I would say 10 years. If we had the ability to do all of the experiments and the facilities to take it forward and things went well, and we, notwithstanding the discussions with regulators, et cetera, that we've talked about, but I'd say we're looking at 10 years. 5 would be an absolute minimum, but I think realistically it would be more like 10 years to come up with something that we could say, potentially this could be deployable. And for us, that would be something that would be targeted to a specific population, probably on an island, probably a fairly small island that's fairly remote and uninhabited, where mice have invaded that island. That was probably the direction and the time frame that we're looking at.
Brian, 10 years or lesser?
Yeah, so we're thinking of 7 to 10 years, maybe in 5 years' time, maybe do some field trials in an island setting or something like that. And then for the technology to be actually fully deployed in the field with, given that there's right regulation, policies and acceptance from the community, I would say 10 years is a acceptable time frame. One other issue to add with regards, because this has come up a lot with regards to suppression of a population. If you look, for example, with mosquitoes, what's being used now, the insecticides, they're killing the population, the mosquitoes. So we're introducing is not any different from what we've been doing for years. We've been suppressing the population, but because we've put the connotation genetic modification, then it becomes very contentious. But what we've been doing all along in vector control is killing the mosquitoes. We've been doing that for years and years with insecticides, with spraying, with environmental management and everything. But what we're trying to say here is that we need complementary tools because these mosquitoes, these vectors are very clever And that's why we're trying to introduce this another technology to complement what's been used right now. Thank you.
Thank you, Brian. I just want to add one thing on the timeline. 10 years if for research, but then for research plus a little bit of regulatory work. But then if the regulations become so sticky and so tedious, then it's also going to increase the duration. So we don't want that to happen, but of course we don't want to compromise on safety as well. So now I'm going to go back to all of you, the experts here. Can you give us the last word? What is your wish? What for Gene Drive, or what would you like to say? Start with Paul.
What would I like to see? I would like to have the capacity to develop what we have further, so that we can actually move it towards the end of the pipeline. As we've all discussed, it's a long journey. So the next step along that journey takes more funding. So we need that. And I'm really interested in developing versions of the technology that we can transfer into other species. So we're not just talking mice, but we can feasibly take into other species as well. That hasn't been really proven possible yet, at least in the vertebrates. Situation is much better in Diptera, but in vertebrates, a transferable generic platform technology that we could move between species would be something that I think would be super valuable. That's what I'd like.
Thank you, Paul. Brian?
I would say I would like for the policymakers, the regulators, to start listening to the scientists and what the research, the results from the research, and they should really stop the fear mongering. There's never, and I've said this before several times, there's never been a technology that is 100% free of any risks. But if the potential benefits of a technology significantly outweigh the risk, then it should, by all means, it should be adopted, consider the safety and efficacy and so forth. If you look for in case of malaria, there's over 600,000 deaths per year. Most of these children and pregnant women are children under 5 years old. So if you're taking the ethical point of view, if you have a technology that could substantially reduce these deaths, then by all means, it should be adopted. Thank you.
Okay, Maha has lost power. So can we continue on with Dr. Nina, please? Your last.
I'm thinking of dengue, and there are many ways to take care of dengue. So we have this integrated vector control. We have this Wolbachia. We're thinking about Gene Drive now. But maybe even before we get into discussions about this, there are so many interventions already, and a vaccine is coming up soon. Of course, we had a problem with the previous one that was actually deployed here, but a better version is coming up soon, and there's a lot of thinking that it will be able to help reduce our dengue cases. So it's choosing which one or looking at all possible options. But I don't know the Philippines if it has the not really the capacity, but the money, the funds to do all that. So still looking at it entirely in its all possibilities.
Thank you. And that'll be for the medical part. How about our environment? How do we deal with it? Last words on that, Ma'am Lita.
Yeah. Okay. So I think our speakers also agree that there might be, and there will be definitely problems with our regulators. And I've always emphasized that I see the potential of Gene Drive to address our problems of invasive alien species. But just the same, there has to be a very good very good interaction between our technical experts and our regulators in the country, especially the regulators, Biosafety Regulatory Committee of our country. And just like GMOs, the introduction of this new technology will have to undergo the same process as GMO did. And I think it was not really very, very smooth sailing during that time. But we have seen that GMOs are, after all, now not a problem anymore. It's just like any kind of hybrid that we have, but the process itself has been very tedious for us at the DNR and the DA. So I think with proper coordination, maybe this can be done. But I'm thinking of 10 years, like you said, 10 years for you to develop the whole process, and only by then can we start the technology. So that is definitely a long process, and our rule says that we have to undergo the same process of producing the gene, and then testing them under containment before it will be field tested and finally released in the environment. So long process, but worth waiting. And thank you, ISAAA, because I learned a lot about gene drive today, and something that I don't really understand so much before I accepted the invitation to talk about this. So that's it. Thank you.
Thank you so much, dear speakers. Hold on a minute. I'm just going to announce that the evaluation form is already in the chat box. Please fill out the forms, the evaluation survey. And yeah, here it is. Here is the QR code. You can just take a picture and respond to the evaluation survey, and you will have your certificates. So we would like to close now our webinar. Actually, we have a lot of questions still unanswered, but many of them are not directly related to Gene Drive. We tried to respond to them as much as we can, but we would like to, please, if you are available, you can send us your questions, and we can send it to our speakers so that we can send it to you, their response. And also, we are also at the ISAAA website. Most of our activities on webinars are posted, and this one is going to be posted there as well. So if you want to replay or listen again to the presentation, you can just go to the ISAAA website and go to the webinar section of the website. So with that, I'd like to thank you all, and I don't have the synthesis that Maha prepared. She was cut off, but it's okay. We're very happy that we have mentioned, or we have presentations from the Philippines on the state of what's the problem, what are the challenges in terms of invasive species. We have the health one from Dr. Gloriani, the environment challenges in terms of invasive species, and yes, Maha was correct that they look good, but they are invasive. We didn't know they were until they got to the Philippines, and they were already wreaking havoc in all of our environment, especially the golden snail, golden apple snail, and fishes, and all that. And so it is very important that we get something, a control mechanism that's going to be sustainable, effective, but then Gene Drive is still going to be, it will take a little bit more time, but acceptance and understanding the mechanism is just one way of letting people know that there is hope that we can control all these invasive species. So with that, thank you very much. And I'd like to recognize the regulators from the Philippines who are also here in our webinar. We are going to have one meeting or webinar with all the regulators exclusively for the Philippine regulators. And you are also invited there. I mean, the regulators, I'm sorry. And it's going to be a one-on-one. So we'd like to hear what your thoughts are, and we would like to help the Philippines in framing policy considerations for Gene Drive, and this will be on November or on December 5.
Okay.
Please register there, those who are regulators in the Philippines. And with that, thank you very much. Thank you all speakers. Thank you all the participants from many different parts of the world. I think my last count was 175. I'm not sure now. There are also people who have raised their hands, but we don't have time anymore. So thank you once again, and we will be waiting for your survey, for the response to your survey. Thank you so much, and have a good day.
Bye-bye.
Thank you.
Bye.